Welcome to the instructional video for version 3.1 of the Beer Spoilage Microtest Kit produced by Spoilage. The kit contains materials for 24 tests. Take an inventory that your kit includes the following: the user instruction booklet, 24 tack beads, 24 lateral flow strips, and vials containing 4 milliliters detection reagent, 1.5 milliliters PCR buffer, and 100 microliters lysis solution. Materials and equipment required but not supplied are detailed in the instruction booklet. The Microbiologique Beer Spoilage Microtest consists of 3 steps. First, there is preparation of samples. Second, there is the PCR analysis. And third, amplicon detection. Sample preparation may be performed using one of 2 mutually exclusive protocols. The enrichment protocol is recommended for clear beer samples and is described in the instruction booklet. The direct detection protocol, using centrifugation, is recommended for clouded, grossly contaminated samples. This is the protocol we will be following in this video. To begin, briefly vortex or swirl the contents of your sample. Then take 1 milliliter of the sample and dispense it into a 1.5 or 2 milliliter polypropylene tube. Be sure to label the tubes appropriately. Centrifuge the samples at 10,000 times G for about 5 to 10 minutes. Take the tube out and carefully pipette out the contents. Make sure at this step you do not disturb the pellet. You will then carefully resuspend the pellet in 50 microliters of distilled water. Vortex your sample briefly to make sure the pellet is mixed well in the water. Take 2 microliters of the sample you just suspended in the water and dispense it into a 0.2 milliliter PCR tube. You will then want to transfer 50 microliters of PCR buffer and 2 microliters of lysis solution from the kit into each of the PCR sample tubes. You will then want to vortex briefly, pulse centrifuge your sample tubes, and then proceed to the PCR analysis step. The PCR analysis step consists of two parts. There is sample lysis and PCR amplification. After resuspending sample in PCR buffer and lysis solution, incubate the tubes in a thermal cycler using the pre-PCR lysis conditions. Using the thermal cycler, you will want to open and run program INC37 to start the lysis reaction. After the run is completed, take your sample tubes out for the PCR amplification step. Recentrifuge the tubes to bring all liquid contents down to the bottom of the tubes. The next step is to transfer the contents into the tack beads. Before transferring any sample, you will want to tap to make sure all of the tack beads are in the bottom of the tube. Next, you will want to transfer 25 microliters of your lysed sample into the tack bead tubes. Again, briefly centrifuge the tubes to bring all liquid contents to the bottom of the tubes. You will then incubate the sample tubes again in the thermal cycler. You will do this by opening and running the BSMT program in the PCR machine to start the PCR amplification. After the run is finished, stop the program, take your tubes out, and proceed to the amplicon detection step. The final step is amplicon detection using DNA lateral flow strips. You will start by centrifuging the amplicon PCR products you just produced to bring all liquid contents down to the bottom of the tube. Next, in a fresh 1.5 milliliter polypropylene tube, you will want to mix 120 microliters of the detection reagent with 10 microliters of the amplicon PCR product you just produced. Next, you will organize your lateral flow strips depending on the number of samples you are testing. Position your lateral flow test strips with the arrows pointing down into the solution. You will then allow the samples to sit for 15 to 20 minutes. Following the run, you will see that a number of bands will appear depending on the spoilage organisms present in your sample. You can position the strip against the internal amplification control line, which allows you to read which organisms correspond to the visible lines. This concludes the demo of the beer spoilage micro test kit produced by Microbiologique. Please refer to your instruction booklet for more information.